question for the NET examination life sciences. Field is the molecular biology. For molecular biology, Leininger book is good, and also we can read Harper and some portion of Karp. Question number 1.
The ATP-dependent RNA-DNA helicase activity is found in Rho protein, sigma factor, CTD, beta subunit of RNA polymerase.
The answer of this question is first, Rho protein. In Rust sarcoma virus genome, the SRC gene encodes a tyrosine specific protein kinase. It affects cell division, cell-cell communication interaction, intramolecular communication, and all. Answer is fourth one, all. Question number 3. Which of the following has helicase activities? TF second A, TF second B, TF second E and TF second F, TF second E and TF second H, TF second H and TF second A. Answer is third one, TF second E and TF second H has these have helicase activities. Question number 4. The incorrect statement regarding RNA replicases. RNA replicase require RNA as the template and will not function with DNA. It lacks a separate proofreading endonuclease activity and has an error rate similar to that of the RNA polymerase. Like the DNA and RNA polymerases, RNA replicase are not specific for the RNA of their own virus. The RNA of the host cell are also replicated. The RNA virus are preferentially replicated in the host cell, which contain many other type of the RNA. Okay, so this is the question based on the Leininger book. Question number 5. Which is the first and most abundant nucleosome constituent to appear on the earth? Adenine, cytosine, uracil and thymine. Answer is the adenine. Question number 6. Which protects the third prime end of the chromosome, making them inaccessible to the nucleases and the enzymes that repair double-stranded breaks? T-loop, methylation, DNA polymerases, homing, retrohoming. Answer is the T-loop. T-loop protects the third prime end of the chromosome from nucleases. Question number 7. After extension of the T and G strand by telomerase, the complementary C and AX strand are synthesized by cellular DNA polymerase, reverse transcriptase, RNA replicase and RNA pol second. It is the cellular DNA polymerase. Question number 8. Which of the following is a ribozyme? RNA is third, RNA is P, RNA is E, all of the above. Answer is the RNA is P. Question number 9. The 5S ribosomal RNA of the most eukaryote is made as a completely separate transcript by pole third, pole second, pole first and third. Answer is the first one, polymerase third. The most common nucleoside modification in eukaryotic tRNA are again conversion of uridine to pseudouridine and adomate dependent nucleus methylation. These reactions rely on RNase, RNSP, SNORNA, protein complex or SNORNase and RNase third. Answer is SNORNA protein complex. Find out correct statement about telomerase. Having synthesized one copy of the repeat, the enzyme repositions to resume extension of the telomerase. After extension of the TG strand by telomerase, the complementary CA strand is synthesized by cellular DNA polymerase starting with on RNA primer. The cellular RNA acts as the template for synthesis of TGY strand for the telomerase. Note the cellular RNA. Telomerase has its own RNA that acts as the primer. Okay, so third one is incorrect. Telomerase synthesis required third prime end of a chromosome as a primer and proceeds in the usual 5' 3' direction. So answer of the previous question, telomerase, is third one. Telomerase synthesis requires a primer and proceeds in usual 5' to 3' direction. This primer is cellular RNA, RNA of telomerase, third prime end of a chromosome, and none of the above. Answer is the third prime chromosome. Question number 13. 30S precursor pre-RNA does not produce 23S RNA, 16S RNA, tRNA, and 5.8 sRNA. Answer is the fourth one, 5.8 sRNA. Question number 14 is missing. Now it is question number 15. Polymerase first transcribed 45S pre-RNA, 30S pre-RNA, 90S pre-RNA, and all. Answer is first, 45S pre-tRNA. Question number 16. This region of the filling the gap acts as the template for the synthesis of TG strand of the telomerase. RNA of telomerase, cellular RNA, third prime end of a chromosome, and replicase. Question number 17. It is a table in the Laniger book, and very good table, and some questions have been asked on this table. TF second A function is to stabilize binding of TF second B and TBP to the promoter, correct? TF second B binds to TBP, recruits pole second TF second B complex. TF second E, it recruits TF second H, has ATPase and helicase activities. TF second F binds tightly to the pole second, binds to TF second B and prevents binding of the pole second to non-specific DNA sequences, correct? TF second H, it unbinds the DNA at promoter, it has helicase activity and phosphorylase pole second, and it recruits nucleotide excision repair protein, TF second H. Okay, so a specific binding is done by TF second F because it prevents binding of pole second to non-specific DNA sequences. So answer is first one. Question number 18. Find out incorrect statement. The reactive oxygen species is a catalytic byproduct of metabolic processes such as respiration and photosynthesis. ROS also act as the signaling molecule regulating plant response to various environmental and endogenous signals. Third one, ROS signals are highly reactive molecules which are generated through the partial oxidation of oxygen. This is doubtful. The majority of the RS are formed in mitochondria and plastid, peroxisome and the cell. Majority of the ROS are formed in mitochondria, correct? Plastid also correct. Peroxisomes and this one also. and answer should be third one, because uh, it is not the partial oxidation, but it is the partial reduction of oxygen. Question number 19. The heterophily is promoted by auxin, cytokinin, gibberellin and abscisic acid. Answer is abscisic acid. Question number 20. ATP binding cassette subfamily G transport auxin, abscisic acid, stigolactones. Answer is B and C. Abscisic acid and stigolactone. It transports these two these two regulators, not auxins. Okay. So for plant physiology book, Tejan Zigar is best book. Tejan Zigar also contribute 40 to 50 marks for this NET examination in different ways, and also Ledinger contributes this one. So minimum, if we depreciate 10 or 20%, then at least 60 to 80 questions are coming directly from these two books. Then if we comments on some of the chapters of Criminology Cube, and then some classical unit 9, so easily there is no problem in preparation or qualifying this examination. Okay, so I think some of the discussion, some of the questions of this discussion will help you in your future examination. Okay, good luck.